Well, thank you. Yeah, so thank you for the invitation. It's a I'm sorry I could not be there in person, but it's a pleasure to talk a bit about our work. And so let me share my video. I'll tell you a bit about some of our recent work and also where I see the field heading in the future of aging, longevity, and genetics. So, okay, so can you see my screen well? Can you see my slides? Yes, with yes, all good.
Yes, of course. We can
Okay,
hear you and
great.
we can see
Well,
your
thank you.
presentation. It's okay.
All right, thank you very much. So yeah, so I mean, again, thank you for the invitation. As a conference on aging and longevity, I suppose I don't need to persuade you of the importance of intervening in aging, and we have a massive social, medical, economic challenge from the aging of the population, and we need to develop ways of intervening in aging and slowing aging, ultimately reversing aging and stopping the aging process. Although I'm not saying that's going to happen anytime soon. Um, and I think this is a really important point to emphasize when you work on aging or longevity, because there's a lot of sometimes opposition to working on aging, and I think we need to make the case that increasing human longevity is ethical. It's the ethical thing to do. Keeping people healthy is healthy, no matter how long. Slowing aging is an ethical thing to do. And so, I've been affiliated with the Blue Hero Oxford Institute on bioethics, and we have a manuscript in preparation to make the case for intervening in aging and extending longevity. To quote, it's the goal, I think, of biomedical research is to allow people to be as healthy as possible for as long as possible. And so I think it's important on aging that, as of us that work on aging and longevity, that we make this case and we make it explicit. And we know that, at least in animals, this is possible. We can extend longevity of animals. We can manipulate aging to a significant degree, in particular at the genetic level. So the first gene identified that modulates aging was H1 by Tom Johnson and colleagues. And now we know that we can manipulate aging with multiple genes and interventions. I'll just briefly focus on genetics, and later I'll talk a little bit about pharmacological interventions as well. But we know the process of aging is plastic, and we can tweak individual genes, and this allows us to let's just forward. And this allows us to modulate aging in animal models. So I've always found this fascinating. And as you mentioned in the introduction, many years ago, we developed this Human Aging Genomic Resources, a collection of databases and tools to help researchers study the genetics and biology of aging. And they've been widely used and very successful. I'll only briefly touch upon one of these databases, which is a GeneAge database of age-related genes, which I think really reflects how much we've advanced in our understanding of the genetic basis of aging. So the current version of the database, which has over 2,000 genes, so in worms now we can tweak individual genes and extend lifespan by up to 10-fold. So I think that's quite remarkable. You have this, worms are microscopic little animals, but instead of living just a few weeks, you can tweak a single gene and allow them to live for more than half a year. And even in mice, we can accelerate aging in mice. So these are two mice of the same age. One on the right is a mutation that's involving DNA damage responses. And the animals not only have a short lifespan, but they're aging faster than normal. And likewise, you can retard aging up to about 50%. So these are two animals of the same age. Again, the one in the bottom has a mutation in growth hormone signaling. And so when you lower growth hormone signal, of course, growth hormone, as the name implies, makes you grow. So when you lower growth hormone signal, you have this very cute, actually, dwarf mice, the small animals that live considerably longer than normal. So I think this is quite remarkable that we can manipulate to some degree aging by genetic manipulations in animal models. And I mean, I invite you to look more at our database. I mean, this is actually done with a colleague in Bucharest, Robbie Tacutu, who now has his own lab in Romania. So despite these advances in modulating aging and manipulate aging in animal models, we still have a very poor understanding of the drivers of aging. I mean, there's many hypotheses, some of that you've heard already today in the conference: oxidative damage, epigenetics, proteostasis, mitochondria, senescent cells, DNA damage, telomeres. So there's many hypotheses for why we age, but we don't yet know. So the empirical evidence is still lacking regarding what are the driving mechanisms of mammalian aging and of human aging. We don't know why we age. But for example, I mean, like I said before, if you manipulate these DNA damage responses in mice, you can accelerate aging, which provides evidence that perhaps DNA damage is a driver of aging, but we don't know for sure. I mean, nobody has made a mouse live 10 years by reducing DNA damage. So we still don't know for sure. Now, one hypothesis I'm particularly interested in, and I'll only briefly touch upon, is this idea that perhaps aging is a programmatic process. I call it the software design flaw. So the idea is very simple, is that perhaps aging is not a result of gradual accumulation of molecular damage. So it's not oxidative damage or telomeres or so on. It's not accumulation of damage, but rather it's a regulated process. Namely, is the programmatic process that is set during development and is regulated by gene regulatory processes set during development. And this actually fits the observation I told you, that if you knock out growth hormone signaling, so if you knock out, for example, growth hormone receptor, you retard aging in mice. So the normal growth hormone receptor is accelerated aging. So you have a gene that is present in mammals that is accelerating aging normally. And if you disrupt it, you retard aging. So this idea of programmatic aging fits from a genetic perspective, because you have what's called wild-type alleles that accelerate aging. And the idea is very simple, is that we have a number of processes, growth and developmental processes, that set trajectories early in life, either changing, for example, a cell type or differentiation or overgrowth or hypergrowth, but then they continue later in life and become detrimental because they're optimized for reproductive ages. But as a form of antagonistic pleiotropy, they continue later in life and then they become detrimental. So one example would be presbyopia, which is the growth of the eye lenses that continually grow, and then they disrupt eye function, and you start to need glasses for reading up close. Time as evolution could be seen as a programmatic process as well, and so on. So it's one hypothesis I'm quite interested in, and for which I think it fits the genetic data we have from animal models. Now, as having said that, there's still a lot of that we don't know. We still don't know what we age, and Sarah already mentioned. We just recently published this paper where we collated 100 open questions in longevity science. So essentially, there's still a lot that we don't know about aging and longevity. Some of these are questions that science has been asking for 100 years or more. Others are newer questions, but there's still a lot that we don't know about the aging process and longevity and its manipulations. But we built this roadmap for geroscience and biogerontology, so the field can focus on what is arguably the most important topics. And this was together with many experts, like Sarah and others that we recently published. Now, I mean, one of the issues is, of course, is translation. And how can we translate findings to humans? We still don't have any proven way of slowing human aging. I mean, we have ways of being healthy. Of course, if you exercise, you become healthier. But whether that slows human aging, whether even that extends lifespan, it's not clear from an empirical perspective. So in spite of many research, big companies studying aging, there is still no proven way of slowing human aging. And I think in terms of interventions or non-genetic interventions, there has been progress, but it seems to be slowing down. So this is an analysis of longevity drugs. So in mice. So this is drugs that extend longevity in mice, specifically from the ITP, the Interventions Testing Program, which is in the US, which is the most robust and quality, actually, an excellent process for testing interventions for longevity in mice. And what you see here is a distribution of the lifespan effects of these compounds. This is average or medium lifespan, and this is maximum lifespan in the bottom. And as you can see, the effect sizes are relatively modest, particularly for maximum lifespan. So the most is 16%, and that's from rapamycin, that we know extend lifespan. But that's the most we can extend lifespan in mice with non-genetic manipulations, which I don't think is great. Okay, it's better than nothing, but I don't think it's great. We still don't know if this will translate to humans, for example. So I think this shows that in terms of longevity interventions, non-genetic longevity interventions in animal models, we still haven't really pushed the boundaries very much. It's still modest effect sizes. And there's various reasons for this. I think in part it goes back to what I was saying earlier about our poor understanding of mechanisms of aging. To give a very quick example, there's been a lot of interest, as I'm sure you're aware, and we've heard about it in this conference today already, in senescent cells and targeting senescent cells. And there's companies trying to develop senolytic drugs that destroy senolytic cells. But actually the biology, I think, may be flawed. Because senescent cells are not really senescence, they're functional cells. And we know that now. We know that actually senescent cells, they play important physiological functions in tissue regeneration, vascular structure, development, wound healing, tissue regeneration, of course, tumor suppression we've known for a long time, even beta cell function in the pancreas. So actually, it turns out that senescent cells are not senescent. I think is an oxymoron to say senescent cells, because they can be detrimental in some contexts. Yes, just like inflammation. I think you equate senescent cell a bit like inflammation. Inflammation can be bad for you, just like senescent cells can be detrimental. But it's also essential, and it's also important, and it also plays functional roles in our bodies. So I think that's a good example of how the biology really is not there yet. Our understanding of the biology of aging is not there, because the challenge now, like I said, senescent cells can be detrimental. So then there will be cases where we want to ablate them, where we want to destroy them. But we need to figure out what those cases. We need to be able to discriminate what are functional senescent cells, which again sounds weird, right? But from dysfunctional or detrimental senescent cells, which we still cannot do yet. And so it's a case of how the biology still needs to progress further. So one of the things that we're doing in trying to identify targets and identify drugs for longevity is employ computational in silico methods for this. So I don't have time to show you all the kinds of methods we've used, but we've done a lot in terms of network biology, machine learning, AI approaches, deep learning as well, to identify new targets and new drugs for longevity. I'll show you one quick example, but we've done quite a lot of work on this, and there'll be more details in our papers. But for example, we're trying to identify new longevity drugs. So what we do is we look at existing longevity drugs, like those from the ITP, for example, and we look at their targets. We look at what pathways they target, look at which processes they're associated with, and then we apply supervised machine learning methods to identify which processes and which targets they have in common, and then try to identify other drugs that have similar targets. So if you have drugs that target, if there's an overrepresentation of a particular receptor, then we'll identify this and we'll be able to then look for other drugs that target the same receptor because they're more likely to be longevity drugs. I mean, there's a lot of statistics behind it, and I don't have time to go into details, but please check out we have a paper on this we published 2 years ago in C. elegans predictions, and now we have another one on mouse predictions as well, mouse longevity drug predictions. But I'll show you one example of success on this, which we publish now. So thanks to this computational drug repurposing approaches, we identified this compound called rilmanidine as a candidate longevity drug. So it's an oral antihypertensive drug. So rilmanidine is used already in the clinic. So it's prescribed to patients with hypertension. And we've done experiments in C. elegans where we showed extends lifespan in both young and old worms. And then, I mean, together with colleagues in Harvard Medical School, Vadim, and also in Zurich, Colin, we've done some experiments in mice. So when mice are fed this compound, it mimics the molecular gene expression changes of other life-extending interventions like rapamycin. So we haven't done a full lifespan study of rilmanidine in mice. That's something we would love to do. But the point is that this is a promising new geroprotector, promising new longevity drug. And because it's already used in the clinic, we may be able to repurpose it for human applications. In other words, trying to do an experimental medicine trial where we look not just at hypertension, but other conditions that maybe it ameliorates. So, I mean, in the last couple of minutes, the other thing we've been doing, so I work with a company called Youth Biotherapeutics, based in the US, which focuses on gene therapies with Yamanaka factors, so partial reprogramming to rejuvenate tissues and restore youthful profiles. So it's a gene therapy approach that we're using, and we've done quite a bit of work on mice. And I mean, recently we received very positive feedback from the FDA in the US to do a first gene therapy with partial reprogramming, with epigenetic rejuvenation gene therapy for Alzheimer's patients. So the idea is to inject a viral vector in the brain of patients, in the hippocampus of patients, with Yamanaka factors, with specific regulatory cassette, and then rejuvenate cells, neurons of the brain, as a potential new treatment for Alzheimer's disease. So that's something we're now fundraising to do, which I appreciate is risky. You know, obviously there's a risk to doing a gene therapy in the brain of patients, but it's been done. There was a recent case done in London, actually, on Huntington's disease, also gene therapy applied directly to the brain. And that, I mean, given how devastating Alzheimer's disease is, and that it doesn't have any effective therapies, this may be a new approach to it. So it's the sort of, you know, ambitious and risky but ambitious, high risk, high reward approach that I think we should be taking. And so, I mean, with that, I told you that aging remains a mystery of biology. We still don't understand well why we age. We don't understand what's the driving mechanisms of aging. There's a number of hypotheses. One hypothesis I'm interested in is the software design flaw hypothesis, that aging is a programmatic process. In other words, it's not damage accumulation, or it's not just damage accumulation. It's not mutually exclusive, but it's not just damage accumulation. These are driven by programmatic processes, gene regulatory processes that probably run on epigenetic level, actually. But it's a regulated programmatic process as opposed to just passive damage accumulation like wear and tear, which I think fits a lot of the observations, for example, in the genetics of aging. And I've told you that we now know of over 2,000 genes in animal models that associated with aging or longevity, which I think is quite remarkable. So in terms of longevity drugs, however, there has been progress. We know of many longevity drugs now. However, it seems to be reaching a ceiling. I mean, in terms of the effects they have, they seem to be relatively modest. They're more modest than genetics. That's not unexpected. But we haven't been able to find, apart from rapamycin, which, by the way, also slows down growth and development, so it fits very well the idea of programmatic aging. But apart from rapamycin, we haven't really discovered drugs that have strong effects on longevity or retarding aging. I told you briefly about our various in silico computational methods to prioritize drugs and targets, which allow us to discover wilmenidine as a new geroprotector, which we're very interested. I mean, if you're interested in collaborating on any of this or wilmenidine, please feel free to reach out. And lastly, I told you the work we're doing with Youth Bio on doing a gene therapy clinical trial for Alzheimer's disease. So with that, I mean, we're based at medical school in Birmingham. These are lab members, the various funders. And I mean, if any of this is of interest, please feel free to drop me an email. Preprints will be available on our lab website and more information about our work. Thank you for your time and attention.